Hey guys, to you I'm an old dude, but my name is Hardy-Weinberg, and I totally stay unbothered because there's literally no drama, no sneaky situationships, i.e. mutations, no new people pulling up, i.e. migration, and no one being thirsty for a specific type, i.e. non-random mating. If everybody's just vibing, finding some more than random, not causing chaos, I stay in my equilibrium era perfectly balanced. That's me, Hardy-Weinberg, and in this lesson, section 7.5, Hardy-Weinberg equilibrium, we're going to be focusing on AP biology or college biology problems. But go, Hardy, before we get started, let's note the disclaimer I listed below. No cap, this is going to be fun.
All right, let's try to figure out the T as it relates to the Hardy-Weinberg equilibrium. Here's the deal. The Hardy-Weinberg equilibrium is a theoretical model. Basically, what we're trying to say is it's creating conditions that really don't exist. It's basically saying, hey, we're going to look at a population, but there's no evolution that's actually occurring here within this population. Like imagine a gene pool just vibing, no changes, no drama, just chilling like a villain. Basically, it's the ultimate tracker for allele frequencies. Nothing wild like mutation or selection is going on. And the Hardy-Weinberg basically tells us what we can expect in our perfect scenario. So if a population is using the Hardy-Weinberg equilibrium, it's literally on cruise control, no cap. There's no genetic flow up or down. Scientists basically use this as a null hypothesis, which is just a fancy way of making sure there's nothing really sketchy going on. Because what we actually do is we take what we receive from the information from the Hardy-Weinberg equilibrium and create that information and compare it to real life scenarios. So we take the statistics from real world scenarios and what we have in our null hypothesis, and we compare and contrast both the data. And the purpose of this is basically if the data basically is so drastically different from what we had in our lab cruise control scenario, we then know then something must have occurred that was kind of shady, like selection or mutation or gene flow. And so that's why this is helpful for AP biology. It really helps identify those evolutionary forces that exist when the Hardy-Weinberg conditions are violated, and allows us to really take a look at genetic stability versus change in a population over a long period of time. So make sure you stick around as we're going to tackle some of these problems together with using some really key indicating equations that you need to know for test day.
Let's review the 5 Hardy-Weinberg conditions. These conditions are important because they allow the Hardy-Weinberg equation to be used when in equilibrium. So the first one is no mutations. Basically, what that means is that when you have these alleles, which are the letter designation, one from one parent and another for another parent, if you have some sort of issue that occurs where maybe one parent's allele didn't pass on or one parent allele was an addition, those are considered some type of mutation. In this case, we're assuming that no new allele will appear due to mutations, and that is allowing for that perfect condition that doesn't allow outside factors to influence the Hardy-Weinberg equation. The next one is no gene flow. Basically, we want to assess what alleles we have within our pool, and we don't want anything coming in or out. Because of this, we're not taking into account gene flow or migration, or immigration or emigration within that population. And that is the second condition. The third condition is no natural selection. The thing is, we assume that all traits are favored equally. So we're assuming that when it's in the pool, there's nothing like a natural disaster that will come in and sort of shake things up. If you remember, you could have in natural selection, for example, a issue where extremes are favored, or you could have issues where intermediates are favored. Both of these conditions are not probable during the Hardy-Weinberg condition because they favor natural selection, and we don't want natural selection. So just to recap, no natural selection is favored due to the idea that there are no traits that are unequally favored. We have all traits that are equally favored, and there are no extreme conditions that exist during that time. The other factor is a large population. Large populations allow for this idea that we have a large pool and we don't have what we call genetic drift. And genetic drift is the tendency to favor one type of gene over a period of time due to the small population that might exist. And in this case, we want that large population to give the most variety with the most stable conditions. And lastly, random mating. There is no mate preference. For example, you don't have tall birds mating with other tall birds. This allows for the most varied results. This naturally, with these 5 conditions, don't exist in the natural environment. So oftentimes you'll be given some sort of equation or problem that allows you to compare the stats that you are given in a natural environment compared to the Hardy-Weinberg condition. So the goal is, if you're given an FRQ or a multiple choice problem, that you understand that the conditions that you're comparing the Hardy-Weinberg to are realistic conditions, and if they vary, then you know that there's some sort of event that happened. And that's what caused the Hardy-Weinberg numbers to be so dissimilar to the natural condition, is that some sort of event occurred that is against one of these conditions. Okay, let's talk about the 2 main formulas that you need to understand. The first formula is P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. These 2 formulas are important to note. Officially, the Hardy-Weinberg principle affiliates with this one. However, let's take a look at the allele frequency equation, which is P plus Q equals 1. Basically, what we're saying is one of the alleles, for example, the dominant allele, plus the recessive allele frequency is added together to give you 1. So P equals the dominant allele frequency, Q equals the recessive allele frequency, and the sum of both of these frequencies always has to equate to 1. Oftentimes, in these types of problems, you're given one or the other. So you're given P, for example, 0.08, and then you have to subtract that from 1 to generate the value of Q. Now, you could use this equation to also solve the Hardy-Weinberg principle. So, for example, let's say they tell you that the frequency of P is 0.8. Then, if you know that the frequency of P is 0.8, then you're going to assume that if you subtract it from 1, that it's going to give you the frequency of Q, which is 0.2. And essentially, what you could do with this information, just by this one number, you can calculate the frequency of homozygous dominant, meaning that there are 2 dominant alleles, so capital A, and that'll give you P squared. Frequency of heterozygous will be 2PQ, so you take 2, for example, times 0.8 times your Q, which is 0.2. And that value alone will give you the frequency of heterozygous genotype. So be aware of that. Sometimes they could ask you just for the frequency of heterozygous genotype. And what they're asking you to do is take both frequencies, the dominant allele and the recessive allele, and multiply it by 2. Then we have the frequency of the homozygous recessive allele, or genotype, I should say, which is in this case, 0.2 squared, because remember, we're taking little a and lowercase a, and that'll be Q squared. We could also calculate it B, and that would be 0.8 8 squared. So the sum of all three of these values will add up to 1 essentially. So essentially you can just have one value and use it with the two equations to calculate the rest of the values. Now let's try to tackle two problems, one being fairly simple and a next one which would be a little bit more advanced. So take a moment, pause the video to read the following question. Once you're done, let's take a look at this together. It says if the frequency of allele A is 0.7, what is the frequency of the allele little a? And they're saying it's going to be what? So we know based on this problem, they give you some parentheses. We're going to ignore that for now. We do know, however, given the equation, that the equation is P plus Q equals 1. We know that P is 0.7, so the frequency of 0.7 plus Q equals 1. We go ahead and subtract 1 from 0.7, that gives us 0.3, so that is the frequency of the little a. Let's try to increase our questions now. All right, so let's take a look at this problem together. They tell you that there's a population of 1,000 individuals that has 360 individuals with genotype AA, capital AA, 480 for genotype heterozygous, and 160 for genotype homozygous recessive. And they're asking you to calculate the allele frequencies. Now remember here, we're given the total alleles, which is the 1,000 times 2. And the reason why we say times 2 is because you're talking about each individual and they get two sets of alleles. So that's why we say 1,000 times 2 gives us the total of 2,000 alleles. Now, knowing this, we have to take into account the A alleles. So we know that we have 360 homozygous dominant alleles. And we know that there's one heterozygous that has one of those alleles that's going to be the capital A. And they're talking about individuals, so it's going to be 480 essentially, because remember they get one of two. So if we're just taking a look at capital letter A, we have the 360 individuals who have big A, so that's going to be 360 times 2 to count for each one of the capital letters. This essentially will be added to our 480. You can say times 1 just because to indicate that it's heterozygous and you're only taking a look at the capital A. And when you put that in your calculator, you're going to end up getting 1,200. Now, if you're taking a look at the recessive allele, you're going to do the same thing with that one, except you're taking a look at the 160 here. So we remember you have 160 times 2 because there's two separate alleles for each individual. And then you're going to count that 480 again. And it's times 1 because they're heterozygous right here. And when you add those two together, you'll end up getting 800. So when they're asking for the frequency of the capital A, remember it's part over total, right? So what's our part here? We have 1,200. What's our total number of alleles? 2,000. And that's going to give us 0.6. If we take a look at the other letter, which is lowercase a, we could do the same thing too, taking into account the lower letter, which is 800, we got, over our total, which is 2,000. And that will give us 0.4. Now, you can double check your answer by saying that you have 0.6 plus 0.4, and that is going to give you 1, which makes sense because remember, P, according to our formula, P plus Q equals 1, right? So our frequency of our our dominant is going to be 0.6, and then our recessive is 0.4. We add them together, that's going to be 1. So that's one way to check it. The other thing, you could solve this problem by saying 1 minus 0.6 gives you 0.4, and also you could go back and double check your answer that way as well to see if you're on the right track. Once again, there are multiple ways to solve this problem, but hopefully that helps you understand these two questions and how you could be asked questions for the AP Biology exam. Make sure to hit that subscribe button, hit that like button, because we hope to continue on with our lessons, especially for this year. Thank you.
Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.